So DELMIC is a company that was founded in 2010 by two professors, Professor Peter Kraut and Professor Albert Polman, at the Technical University of Delft and the Armhoff Institute in Amsterdam, both in the Netherlands. And we develop, as Pam said, integrated products for cathodoluminescence and correlative microscopy. So what is correlative microscopy? In order to go into that, let's look at two popularly used microscopy techniques. The first is fluorescence microscopy. So a little bit about the phenomenon of fluorescence. When an atom or a molecule in ground state, which is shown by the blue dot here, when you shine light of a certain wavelength on this, it can absorb photon and go into an excited state, which is shown in orange. And from there, it can decay to a lower energy state, shown in red. And the difference in energy can be released as a photon, which is the phenomenon of fluorescence. Fluorescence microscopy is a technique commonly used in biology, for example, where biological specimens can be tagged with specific antibodies, and these antibodies can in turn be tagged to fluorophores, which are molecules that emit fluorescence. And by imaging these molecules in a microscope, you can essentially image different parts of the biological specimen. So the labeling with fluorophores in this case is then specific, because the antibodies are specifically tagging to different parts of the cell. This gives the advantage of functional information, of obtaining functional information from this microscopy technique. And the resolution is of the order of 200 nanometers, which is the diffraction limit of light. Another technique, complementary to this, is electron microscopy, where a biological specimen is placed in an electron microscope, a scanning or transmission electron microscope, and a high-energy electron beam, which is shown in this image as primary electrons, is made incident on the sample. Since it's high energy, it penetrates to some depth within the sample, and it generates a whole host of scattering events. So this causes backscattered electrons, Auger electrons, secondary electrons, and other effects like X-rays, and transmitted electrons to be generated in the sample. By labeling a sample with heavy metals, which is then a non-specific labeling because it attaches to a lot of parts within the biological specimen, it's possible to generate contrast in an electron microscope so that we receive structural information about the sample. The resolution in electron microscopy is of the order of 5 nanometers, sometimes better. And correlative microscopy then is a technique that combines the two. So it's a combination of fluorescence and electron microscopy. And the advantages are that you can receive both functional and structural information, one from fluorescence microscopy and the other from electron microscopy. And this leads to a very nice result, which is that you can image unknown ultrastructure. Because by using fluorescence microscopy alone, it's only possible to see what you have chosen to see. That is to say, by tagging the biological specimen with certain antibodies, you choose what will be imaged. In electron microscopy, on the other hand, everything that is there is seen. So correlative microscopy is a technique that combines the advantages of the two, because it's also possible to have long-range labeling, multicolor labeling for fluorescence microscopy by tagging the biological specimen with different fluorophores. And it leads to unbiased identification of cells, because by using electron microscopy alone, you are restricted to identifying cells based on their structure or morphology alone, which can lead to an error in the identification. But using a combination now of structure and function, it's possible to identify cells much more accurately. And one more important aspect of this is that it allows for liquid cell imaging, which is very important for biological applications. So in a nutshell, how does correlative microscopy work? This is, in green, you saw a fluorescence image, and the black and white contrast is generated from an electron image. And the resultant image that you see now is an overlay of the two. So you can see functional information in green in the context of cellular ultrastructure in grayscale from the electron image. So on the left, you see an overlaid electron and fluorescence image. And on the right, you can see another overlaid image with multicolor labeling. So in blue and in orange, you can see different parts of the cell have been identified. And once again, in context of the cellular ultrastructure. So at this point, I'd like to just pause and ask if there are any questions. If not, we can proceed to looking at the SECOM platform, which is a platform on which integrated electron microscopy and fluorescence microscopy are being developed for simultaneous imaging. The image on the left is what the integrated SECOM system on a microscope looks like. In blue is the cover of the SECOM module, which is mounted on a standard scanning electron microscope here. And on the right image, you can see a schematic of how the system works. If you can see the pointer on your screen, the orange disk shows the sample of interest, which is placed in vacuum in the electron microscope. In green, you can see the electron beam being focused on the sample. It generates contrast, which can be detected by the secondary electron detector here, or a backscatter detector here, or a variety of detectors. Simultaneously with that, the light source shines light on the sample, which is then taken by means of the dichroic and the mirror, and it excites fluorescence in the sample. This fluorescence is then collected back on the camera. So this is the schematic for acquisition of electron and fluorescence image on the SECOM. So the main aspects of this integrated microscope are that the workflow is streamlined. So it's no longer needed to prepare the sample for fluorescence microscopy, take it to a fluorescence microscope, take an image, take it out, then go to an electron microscope, take an image, and then overlay them manually. This workflow is streamlined and is done in situ in this system. It's also possible to retain the high NA fluorescence with the objective. The overlay procedure between the electron and fluorescence images is now automated. And this SECOM is something that can be retrofitted onto existing scanning electron microscopes. And there is an open source software for acquisition and analysis. So we will soon go into each of these aspects in detail. But let's take a look at how the SECOM itself looks like on the inside. So in the large image on the left, the annular metal disc that I'm showing here is the sample holder. The sample is placed on the cover glass. If there is an ITO slide with glass on it, it can be glued onto this in a stable way. This large base plate is the sample stage, which can move in X and Y. And here is the XYZ stage for moving the objective lens. The image here on the right shows you an objective lens with the droplet of oil in between the sample and the objective. So this system is compatible with immersion oil objectives, and it can be used in vacuum. And as I mentioned before, the SECOM is a retrofit compatible with a host of microscopes like FEI, Zeiss, Tescan, Hitachi, and JO. So now, coming back to the features of the SECOM, the automated overlay is one of the most important aspects. So how this is done is, in principle, using cathodoluminescence. So when the electron beam is incident on the glass, it generates cathodoluminescence there. And this cathodoluminescence, or CL, can be detected by the camera when it is switched on. So using this electron beam impingement, it can write many spots. And each of these spots then, you can imagine, acts as a temporary fiducial marker. So by writing such a grid and detecting the cathodoluminescence that comes from it, it's possible to correlate the electron and the fluorescence images. This correlation technique it's independent of the sample, it's fully automatic, and it's very precise. And the things that this method can correct for in the overlay are rotation, scaling, and position or translation. So the acquisition and image overlay are then performed using this method that I just described. So the first step is to acquire a fluorescence image. That's the image on the left, the bottom left that you see. After this, the sample has the electron beam scanned over it. So the fluorescence image is acquired by shining light on the sample in one go. And the electron image is acquired by scanning the electron beam over the region of interest. And subsequently, the grid is patterned using the electron beam for the pointer alignment. This entire procedure of acquisition and overlay is automated. And the software that is used for performing the imaging is called DELMIC. It's an open source software. It can be used to navigate over the sample and control the electron microscope and the fluorescence microscope. And it has a very user-friendly graphical interface. And it also supports a full scripting interface. So you can write your own script for control and automation. The features of the software that are relevant here are the automated alignment procedure that I mentioned. There is also, in addition to the live mode, there is an image acquire mode for the fluorescence and electron microscope, which you can see as a blue button here, which performs the whole procedure in an automated fashion. It's possible to acquire multiple color channels, which is one of the main advantages of fluorescence microscopy. So what you see here is one of the streams. So you can excite a certain fluorophore. You can add streams, excite different fluorophores, and then overlay them individually with the electron image. And all of this is simultaneous, so it's real in situ. And you can directly visualize the overlay. Here you see a large field of view fluorescence image, and in the center you can see a high magnification, high resolution electron image. And by operating the slider here, you can change the mixing ratio of the electron contrast and the fluorescence contrast. So you can visualize the overlay as you wish. And this supports different file formats like OME-TIFF and HDF5, which are used for saving large data sets. So now that brings us to some applications of the SECOM system. One of the important applications that we are working on is in the field of neuroscience. So electron microscopy is capable of high-resolution imaging of synaptic connections in the brain. This is one of the main fields of research in neuroscience. But the disadvantage of electron microscopy here is that you need a long imaging time per pixel, because it's not a one-shot technique. It's not a one-shot illumination, but it requires scanning of the E-beam pixel by pixel across the whole image. Fluorescence microscopy, on the other hand, is capable of imaging a large area because it's fast. So in principle, you can attach labels to the neurons, fluorescent tracers, and image the brain connectivity using fluorescence microscopy. But in that case, the disadvantage is the insufficient resolution because it's diffraction limited. So using correlative imaging, it's possible to have a large field of view fluorescence microscope image and a high magnification, high resolution electron image overlaid. So you can have both kinds of information of the same region. And on the right here, you can see what a resultant image might look like. This is an example of a correlative image of neural connections inside the brain of the songbird. The green spots that you see here are the fluorescence from the neural tracers, visualized in context of the ultrastructure of the brain, using contrast from the electron microscope. A couple of important points here, that issues that come up in fluorescence microscopy versus electron microscopy. And it's relevant here because we are doing the two simultaneously. So fluorescence microscopy usually requires or often requires water to be present in the system in order for the molecules to fluoresce, for their fluorescence action to be retained. But electron microscopy, because it requires vacuum, needs samples to be dry. Another issue that comes up, a conflicting issue often, is the heavy metal staining that's used for generating contrast in the electron microscope. This can lead to quenching of the fluorophore so that it no longer gives out the fluorescence emission. So in this example, which I showed here, in this image that was acquired, there was a labeling protocol that was used, which involved electron staining, heavy metal staining for electron imaging, followed by on-section immunolabeling, which was successful in retaining the fluorescence. So that's why it was, there are protocols which are optimized for obtaining a correlative image. The next application that I'd like to discuss is in the research of type 1 diabetes, or diabetes mellitus. So this is a disease in which the beta cells in the body, which produce insulin, are attacked and destroyed. So beta cells are present in the pancreas, in a cluster of cells called the islet of Langerhans, and they are responsible for detecting the sugar level in the blood. If the sugar level is elevated, for example, immediately after you've had a meal, then the beta cells sense this, they release insulin, and they signal for the cells in the body to uptake the sugar and convert it to energy. So in this autoimmune disease, type 1 diabetes, the beta cells are no longer able to perform this function. So what happens is that the sugar remains in the blood, and the cells can die from lack of oxygen. So it's a life-threatening disease which can lead to multiple organ failure and cell death. The mechanism for type 1 diabetes is not well understood. So if we want to image the changes taking place in the body during diabetes, the question is what imaging technique is optimal. So if you have a fluorescence image like this one, it's possible to have functional information because different parts are tagged specifically, but the resolution, as you can see, is quite poor. It's commonly used technique. Electron microscopy is a commonly used technique for imaging, which is high resolution, but the disadvantage is that it's labor intensive and is also error prone as far as identification of cells is concerned, because the identification is performed based on the morphology alone. Using correlative imaging, it's possible to get functional information, so it's possible to image the beta cells specifically in the context of the cellular ultrastructure. So the image that you see here is a simultaneously acquired correlative image of the islets of Langerhans in the pancreas. It's an overlay of the fluorescence image where the insulin or the beta cells are labeled in orange. The green is the guanine quadruplex, and in blue is the nucleus. And this is overlaid on a scanning electron microscope image. So this is an SEM image acquired on the SECOM. And a few points to note here about the sample preparation, which is very relevant, is that this is an image of thin sections of healthy rat pancreas, so 80 nanometer thick sections. And the procedure was fixation followed by osmium tetroxide treatment, and then dehydration, embedding in resin. And the final step was the immunolabeling or the fluorescence labeling. So you can see that it's possible to get functional information as well as very high resolution structural information in one shot. If there are any questions at this point, I'd like to stop and take a look. And if not, we will proceed to one of the applications that we have recently started working on, which is in the field of medical diagnostics, in particular in pathology, which is a study of disease based on analysis of the fluids in the body. That's performed in a laboratory then, and usually in hospitals. So the current workflow is fluorescence image followed by transmission electron image. So the disadvantage here is that TEM imaging, or transmission electron imaging, is very expensive. It's time consuming. It's also usually present at a completely separate facility, so it's not simultaneously imaged with the fluorescence, and which then leads to the challenge that it's difficult to locate the high resolution features or particles in the fluorescence image, which are indicative of the disease, since it's not simultaneous. For this application, the DELPHI, which is a tabletop system for correlative imaging, shown here. So on the right, you see a computer screen and a keyboard, and on the left is the DELPHI, the tabletop correlative imaging system, the entire system. It's very compact. And the main advantage is that it's very fast and easy to use. So you can have a simultaneous fluorescence and electron image after a pump down time of just 5 minutes. And it's very nice because it's easy to use, it's very user-friendly, it doesn't require experienced or highly trained users. So for applications in hospitals where they need to go through patient samples rapidly, you need to have a system which is fast and reliable, which is what the DELPHI can do. So an example of the application of the SECOM for pathology is you can see here. This is a fluorescence image of a human kidney section, of a part of it. And the feature that you see here is a glomerulus. And this, on the DELPHI, a simultaneous acquired image is shown on the right. So the SEM image on the right was acquired simultaneously with the fluorescence image on the left. And from the electron contrast, it's possible to see the structural information that is necessary to understand the functional information that you see on the left. So if there are particles or features of interest present in this region, they can be visualized at higher resolution in the SEM image using a backscatter detector on the DELPHI. One of the highly specialized applications possible with the SECOM has been developed into a special super resolution system. So it's a super resolution microscope developed based on the SECOM, which allows you to carry out CLEM, simultaneous correlative light electron microscopy in vacuum. In this system, there is a special workflow. So the sample is HeLa cells, which have been infected with virus and then frozen and embedded in resin. 200 nanometer thick sections were cut and mounted in the integrated setup in the super resolution microscope. So the sample holder that you see here is the same one that I showed on the standard system. And the sections are cut and placed on an IPO cover slip and mounted on this holder. This holder then fits onto the super resolution system. So the procedure is the fluorescent cells are located using the SECOM platform in the ordinary wide field mode. So an image such as this is acquired. And then there is a laser in the system for the excitation, and the power of the laser is increased, which causes the fluorophores to blink. And approximately 30,000 images are collected, which is the super resolution mode of the system. And this is the resultant image in super resolution. Subsequently, the electron microscope image is acquired using a backscatter electron detector. And this is then the EM mode, which gives you a high resolution image such as this. And the same sequence, so wide field super resolution EM, is performed over several locations in XY or in Z, leading to a stack of images like this. So this is the wide field image in the first region of interest, then move over to another region of interest in a wide field image. And these are the super resolution images corresponding to those. And the bottom row is the scanning electron microscope image corresponding to this. The super resolution images are then reconstructed using Thunderstorm, which is a software, and overlaid. So there is the overlay of the wide field EM and super resolution EM that you see here, again, is a tile of images. And zooming in, if we look at a high resolution of one of these, you can see clearly, if you compare the resolution in the wide field image of the fluorescence to the green spots with the super resolution mode, you can see the clear enhancement in resolution, optical resolution. In fact, the resolution is 85.5 nanometers. So this is a super resolution system based on the SECOM platform. And that brings me to a project that we are currently working on, which is for large area imaging. In biology, it's important to have an understanding of networks and connectivity over a large sample area. And for this, we are developing an integrated workflow, which includes automated stage movement, automated alignment, as well as stitching. So the goal is to be able to achieve, in an automated fashion, something like this. So an electron image in one location, stage move, another image, stage move, another image, and in this way acquire a pile of images which would be stitched together. And the simultaneously acquired fluorescence image should be overlaid on this to have a large area correlative image acquired simultaneously and stitched together. So this is the correlative image that you see. So essentially, the SECOM is an integrated correlative light and electron microscope, which enables you to do correlative microscopy very fast, with a high optical quality and very accurate overlay. That gives you the advantage of being able to spend your time acquiring data instead of aligning the fluorescence and electron microscopes. The overlay procedure, as I said, is very accurate and reliable. So it also offers the possibility to use vacuum-compatible immersion oil for high NA fluorescence. And the SECOM is compatible with almost all SEMs. So there's also support for beam deceleration mode, immersion mode in the SEM, and for using a variety of detectors like the secondary, backscattered, EDX, and in-lens detectors. So that brings us to the end of this webinar. This was presented together with our partners, AXT, and I would like to stop, and if there are any questions, I'm happy to answer them.